There's a fish sitting on the bottom at McMurdo Sound in Antarctica, about 400 feet down, and it's resting against ice. Not near ice, against it. Crystals of frozen seawater are touching its gills, drifting through the water column around its head, sliding along its skin. The water it's breathing sits at roughly 28.5 degrees Fahrenheit, which is below the point where fresh water turns solid, and which is also, and this is the part that matters, below the temperature at which that fish's own blood should turn to slush. Take a sample of its serum, put it in a tube, cool it down. The salts and sugars and proteins dissolved in it push the freezing point down a little, the same way road salt does, and you land somewhere around 30 degrees Fahrenheit. That's where the blood of a fish like that ought to freeze. And the ocean it lives in is about 2 degrees colder than that. Every second of its life, it's sitting in water cold enough to seed ice inside its own circulatory system, in physical contact with the very crystals that would do it. It's been doing this for millions of years. Its ancestors have been doing it since before the ice sheets finished forming. So something in that fish is cheating. And when Arthur DeVries went down to McMurdo in the late 1960s and started pulling these animals up through holes in the ice to find out what, the answer turned out to be none of the obvious candidates. Extra salt in the blood would work in principle, and there's no extra salt worth mentioning. A thicker skin would slow the whole process down, and the skin is unremarkable. Behavior would solve it if there were warm patches to hide in, and there are no warm patches. What he found instead was a molecule nobody had a category for. A specific, weird, sugar-decorated protein that grabs ice crystals by the face and refuses to let them grow. That is one system. There are a lot more, and once you start looking for them, the ocean stops being scenery and turns into something closer to a torture chamber with very specific instruments in it. Because here's what I think gets missed in almost every conversation about marine animals. We talk about them as though the ocean is one place. It isn't. It's a stack of completely separate physical insults layered on top of each other, and an animal that solves one of them is usually helpless against the next. Cold is one insult. Pressure is a different one, with a different mechanism of harm, doing damage in a way that has nothing to do with temperature. Running out of oxygen is a third. Salt pulling water out of your cells or shoving water into them, depending on which direction you crossed, is a fourth. Ultraviolet light frying the chemistry of your skin near the surface is a fifth. Heat, actual scalding heat, at the exact bottom of the sea where you'd swear heat has no business being, is a sixth. And every single one of those has produced a piece of biological hardware that a human body simply doesn't have. There's a blank where the organ or the molecule or the reflex ought to sit. I want to walk down through those insults one at a time, and each time I want to start with what that insult does to a person. Not in a gruesome way, in a mechanical way. What breaks first, what the body tries to do about it, and precisely where the attempt runs out of road. Because the comparison in the title only means something if you know what the failure actually looks like from the inside. Saying an animal survives cold water is a greeting card. Saying that a human being dropped into the same water loses control of their breathing inside 3 seconds, loses the use of their hands inside 10 minutes, and that no amount of training changes the underlying chemistry tells you what the fish is actually pulling off. Start with the cold then, because it's the one people think they understand. Here's what water at that temperature does to you, and it isn't what most people picture. Most people picture hypothermia, a slow cooling, shivering, then confusion, then the drift. That's real, but it's almost never what kills first. The first thing that happens is a reflex called cold shock, and it fires within a second or two of your skin hitting the water, long before your core temperature has moved at all. Cold receptors in your skin dump a signal into your brainstem. You gasp, involuntarily, deeply, one enormous inhalation that you can't stop and can't delay. And if your head happens to be underwater at that moment, that gasp is a lungful of seawater. Then comes hyperventilation, and the numbers on this are worth hearing. Researchers who have measured it, including Mike Tipton's group at the University of Portsmouth, have recorded ventilation rates several times above resting, in some cases many hundreds of % above, which blows off carbon dioxide so fast that the chemistry of your blood swings alkaline. That causes constriction of the vessels feeding your brain, dizziness, tunnel vision, confusion at the exact moment you need to be solving a problem. Your heart rate spikes and your blood pressure spikes at the same time because every vessel near your skin has clamped down to protect your core. The safety world compresses this into a rule of thumb they call 1-10-1. 1 minute to get your breathing under control. 10 minutes of useful movement before your hands and arms stop obeying you, because cooled nerve and muscle simply conduct and contract more slowly, and there's no willpower fix for a nerve that's gotten cold. Then, roughly 1 hour before the core drop becomes life-threatening, assuming you're still at the surface. Notice what's missing from that list. At no point in that entire sequence does the human body do anything to lower its own freezing point. The question never even comes up because we die of the cold long before we get anywhere near freezing solid. Our defenses are all about slowing heat loss and protecting the core. Insulation, vasoconstriction, shivering to generate heat, and if you happen to be cold adapted, a bit more brown fat and a slightly better shiver. Every one of those is a way of managing heat. The chemistry of the blood itself never changes at all. That's the fork in the road. Somewhere back in the evolutionary history of Antarctic fish, the water stopped being a thing you could manage thermally. You can't insulate your way out of it, because a fish is the same temperature as the water by definition. It has no core to protect. There is no inside. The ocean is inside it. So the only remaining move is at the level of the molecule. There's an obvious way to lower the freezing point of a liquid, and you already know it, because you've watched a highway crew do it. Dissolve stuff in the water, salt, sugar, anything. The dissolved particles get in the way of water molecules trying to line up into a crystal lattice, and the more particles you cram in, the lower the temperature has to go before the lattice can form. Chemists call this a colligative effect, which just means it depends on how many particles you have, and not at all on what kind they are. 1,000 sodium ions and 1,000 sugar molecules do exactly the same amount of work. That's the route evolution didn't take, and the reason is arithmetic. The blood of a typical marine fish carries somewhere around 1/3 to 1/2 of the dissolved load that seawater does. To push its freezing point down to match Antarctic seawater by brute chemistry, a fish would need to roughly double or triple the concentration of salts in its blood, which means every cell in its body would be sitting in a solution that's pulling water out of it constantly. Enzymes stop working, proteins fall apart. You have solved the freezing problem by replacing it with a slow chemical dehydration that kills you just as thoroughly, only with better paperwork. What the notothenioid fishes of the Southern Ocean evolved instead is one of the more elegant pieces of chemistry in the animal kingdom, and it works on a completely different principle. The molecule is called an antifreeze glycoprotein. Structurally, it's almost insultingly simple. A repeating unit of 3 amino acids, alanine, threonine, chained end to end, with a little 2 sugar tail hanging off every threonine. Repeat that unit anywhere from 4 times to 50 times, and you get the whole family, 8 different sizes of essentially the same thing. There's no fancy catalytic pocket, no active site, none of the Baroque folding you see in an enzyme. It's a stick with sugars on it, and it doesn't lower the freezing point of the blood in the ordinary sense at all. Here's the trick, and once you see it, the whole thing clicks. Ice does get inside these fish. That surprised people. The fish aren't sealed. Crystals come in through the gills, through the skin, and they swallow them with seawater. The animals are essentially full of tiny ice fragments at all times. Freezing to death for one of these fish comes down to what the ice does once it's already in there. Keeping it out was never on the table. A crystal grows by adding water molecules to its surface, layer by layer, at the growth steps along the edges. What the antifreeze glycoprotein does is stick to that surface, irreversibly. It parks itself on the ice, and once it's there, it doesn't come off. Now imagine a few thousand of those parked all over the crystal face. The ice can still add water molecules, but only in the gaps between them, which means the growing front has to bulge outward through each little window like dough through a grill. And curved ice is unstable ice. This is a real, measurable piece of physics, the Gibbs-Thompson effect, sometimes called the Kelvin effect. A curved surface has more surface energy per unit of volume than a flat one, so a bulging front of ice needs a colder temperature to keep growing than a flat sheet does. The tighter you force the curve, the colder it has to get. Raymond and DeVries laid this out as the adsorption-inhibition model back in 1977, and the core of it's held up through decades of increasingly nasty experiments. The result is a gap. The temperature at which the fish's blood melts stays roughly where the salt content says it should. The temperature at which it freezes drops well below that. Two different numbers for what's normally one number, and the space between them is called thermal hysteresis, which is a mouthful for a very simple idea. The ice inside that fish is stuck. It can't grow, and it also can't quite melt. And we'll get to why the second half of that sentence is a problem later on. What I find hard to get over is the efficiency. Per molecule, this thing is hundreds of times better at holding off ice than salt is. A fish doesn't need to poison its own cells with solute. It needs a modest amount of a protein that spends its entire career doing one job, which is standing on ice and saying no. There's also a lovely detail about where it gets made. For a long time, everyone assumed the liver, because the liver makes most of the things floating in blood. It turns out a huge share of it's manufactured in the exocrine pancreas and squirted directly into the gut, which makes complete sense the moment you remember the swallowing problem. These fish drink seawater. Seawater at that temperature carries ice crystals. If the antifreeze only lived in the bloodstream, every mouthful would be seeding a fresh crystal in the digestive tract, on the wrong side of the wall, where the blood supply can't reach it. So the fish coats its own intestinal contents. It's putting antifreeze in the tank and in the fuel line at the same time. You have probably eaten a distant relative of this molecule, incidentally. When food scientists went looking for a way to keep low-fat ice cream from turning grainy in the freezer, the problem they were solving was exactly the fish's problem, which is small ice crystals slowly merging into big ones. The industrial answer was an ice-structuring protein, grown in yeast, working on the same adsorption principle that DeVries described in Antarctic fish blood. Somebody looked at a molecule evolved to keep a cod-like animal alive under a sheet of polar ice and thought, yes, this will improve dessert. Improve. I have complicated feelings about that, and none of them are negative. Now flip it around and ask what actual freezing does to human tissue, because the mechanism is worth knowing, and it explains why we have never evolved anything remotely like this. When your finger freezes, the first ice doesn't form inside your cells. It forms outside them, in the fluid between cells, because that fluid is slightly less loaded with dissolved material, and so it crystallizes first. As soon as pure water starts locking itself into ice out there, everything that was dissolved in that fluid gets concentrated into the shrinking pool of liquid that remains. Now the outside of your cells is saltier than the inside. Water starts leaving the cells to balance it. The cells shrivel. Proteins that were happily folded in a normal salt environment find themselves in a brine, and they start to come apart. Membranes, which are held together partly by water, get chemically wrecked while the cell dehydrates. If the freeze happens fast enough that water can't escape in time, you get the other failure mode, which is ice forming inside the cell. That one is mechanical, and it's final. And then, the thaw does its own damage. Blood vessels that were frozen leak when they reopen. Tiny clots form. Inflammatory chemistry floods the tissue. And a great deal of what people call frostbite injury is actually reperfusion injury happening hours after the rewarming, which is why the medical guidance is so specific about not thawing a frozen extremity if there is any chance it will refreeze on the way to help, and about getting it into professional care rather than improvising. That's real clinical guidance with a lot of decision branches behind it. And it isn't the point of this video, so take it to a doctor and not to me. The point is that our entire relationship with ice is damage control after the fact. We have no molecule that watches for the first crystal. People have tried to borrow the fishes. Cryobiologists have been putting antifreeze proteins into organ preservation solutions for decades, and the results are honestly mixed in a way that's instructive. At low concentrations, they help. Push the concentration up and something perverse happens. The protein binds to some faces of the crystal and not others. So instead of stopping ice, it shapes it. And the ice grows out in long needles along the unblocked direction. Needles are worse than blobs. The molecule that saves the fish can spear the cells of an organ you were trying to protect because the whole system was tuned by evolution for one specific animal, at one specific temperature, in one specific fluid, and it doesn't port cleanly. Where the fish version came from is, to me, the best part of the story. In 1997, Liang Biao-Chen, Arthur de Vries, and Christina Cheng traced the notothenoid antifreeze gene back to its ancestor and found a digestive enzyme, trypsinogen, the precursor of a protease, an ordinary gut protein whose day job is chopping up food. A small stretch of that gene, 9 nucleotides long, coding for that alanine-alanine threonine unit, got copied and recopied dozens of times, and the flanking machinery of the old enzyme was largely discarded. Evolution didn't design an antifreeze. It found a stutter in a pancreas gene and let the stutter run, which, incidentally, explains the pancreas thing from earlier rather neatly. The gene never left home. And then it happened again, on the other side of the planet, in a completely unrelated animal. Arctic cod carry an antifreeze glycoprotein with essentially the same repeating structure, and it didn't come from a trypsinogen gene. Different genetic ancestry, same answer, because when you need to stop ice from growing and you're working with amino acids, there appear to be very few good solutions, and physics keeps pushing organisms toward the same one. The southern version is the older of the two, tied to the cooling of the Southern Ocean over the last several million years, and the northern one looks considerably younger, which lines up with the North freezing later. Two independent inventions of the same tool, at two different times, for the same reason. But there's a bill, and it took until 2014 for anyone to notice it. A team including Paul Chico, Christina Cheng, and de Vries went looking at what happens to those internal ice crystals when the water warms up in the Antarctic summer. Logically, they should melt. The melting point of the fish's fluids is higher than the freezing point, remember, and summer water at McMurdo does creep up, so the ice should go away. It doesn't go away. The antifreeze proteins that are welded to the crystal surface also block melting. The ice sits there superheated, above the temperature where it has any business existing as a solid, held together by the same molecules that were keeping it small. The team found crystals in wild fish caught in the warmer part of the year, in the blood, and concentrated in the spleen, which appears to be acting as a sort of collection bin for the things. Their long-term temperature record of the habitat suggests summer never reliably gets warm enough or stays warm enough to clear them. So the working picture is that these fish accumulate ice inside their bodies over a lifetime and never fully get rid of it. What that cost them over decades, nobody knows for certain yet. And I'd rather say that plainly than pretend the story ends in a tidy victory. It's the first case anyone has documented of superheated ice occurring in nature. and it exists because a solution that good has an edge to it. That's the shape of the whole subject, actually. Every system in this video is a trade. It's just that some of the trades are more visible than others. Cold, though, has a second property that has nothing to do with freezing, and it sets up the next problem completely. Cold water holds more dissolved gas than warm water. Chill the Southern Ocean down to the point where it's about to turn solid, and you get seawater carrying more oxygen per gallon than almost anywhere else on the planet. At the same time, everything living in that water has a metabolism slowed to a crawl by the temperature, so oxygen demand is unusually low. High supply, low demand. Hold those two facts together, and something becomes possible that shouldn't be possible in a vertebrate. In 1928, a young Norwegian biologist named Ditlev Rustad was working in the far Southern Ocean when he pulled up a fish he didn't recognize. Long, pale, big-jawed, unpleasant-looking, in the way that deep cold fish often are. The whalers apparently called things like it white crocodile fish. What stopped him was the blood. He cut into the animal, and what came out was colorless. Not pale red, not pink, clear, like slightly cloudy water. He wrote it down. Nobody did much with the observation for a quarter of a century, until Johann Rude published a proper account in Nature in 1954, and the physiology community had to sit up and deal with what it meant. Here's what it means. The ice fishes, the family Channictidae, 16 or so species of them living around Antarctica, are the only vertebrates on Earth that go through adult life without hemoglobin. The gene is there in the genome as a wreck. The protein isn't made. There are essentially no red blood cells in the circulation to speak of, and the oxygen those animals run on is dissolved directly in the plasma, the way sugar is dissolved in tea. To understand how insane that is, you need one number about your own blood. Take 100 milliliters of your arterial blood. It's carrying roughly 20 milliliters of oxygen. Of that 20, about 0.3 of a milliliter is simply dissolved in the plasma. Everything else, all 19.7 of it, is locked onto hemoglobin inside your red cells. Hemoglobin is doing essentially all of that work. The dissolved fraction is a rounding error that medicine mostly ignores. The ice fish runs on the rounding error. Measured properly, ice fish blood carries less than 1/10 of the oxygen that the blood of its red-blooded Antarctic cousins does. If you scaled that to a person, you'd be talking about a hemoglobin concentration somewhere around 1.5 grams per deciliter. For reference, a healthy adult sits somewhere in the 13 to 17 range. Hospitals start having serious conversations about transfusion in the 7s. Below 5, the heart is in trouble. There's a scattering of case reports of people surviving at numbers under 2, and every one of them is a medical emergency with a helicopter involved. So the obvious question is how an animal does that as a lifestyle, rather than as a crisis, and the answer has two halves. Half of it's the environment doing the work, and half of it's the most aggressive cardiovascular remodeling job in the vertebrate world. The environmental half we already set up. Water at the freezing point of seawater holds a lot of dissolved oxygen, considerably more than warm tropical water does, and the Southern Ocean around Antarctica is about as oxygen-rich as seawater gets. Meanwhile, the fish itself is running a metabolism at roughly 30 degrees Fahrenheit, which is to say barely running at all. Chemical reactions slow down as things get colder. An animal at that temperature needs a fraction of the oxygen per pound that a warm animal does. That covers the supply and demand. It doesn't cover the delivery, and delivery is where the engineering shows up. Ice fish hearts are enormous. Compared with related fish of the same body size, the ventricle can be several times heavier. And against typical bottom-dwelling fish, generally, the relative mass runs up to about 10 times larger. It's also thin-walled, built to move a huge volume of blood at low pressure rather than a modest volume at high pressure, which is the exact opposite of the design philosophy in your chest. Weight for weight, The amount of blood these animals push per minute is 4 to 5 times what a red-blooded fish of the same size pushes. Then there's the amount of blood. Mass-specific blood volume in ice fish runs 2 to 4 times higher than in ordinary bony fish. More fluid, moving faster, through vessels that are also wider. The capillaries are notably large bore. The vascular beds are dense, and because there are almost no cells suspended in it, the blood is thin, which matters enormously in the cold, since cold blood full of red cells would be sludge. Losing the red cells actually solved a viscosity problem that the cold had created. They also breathe through their skin. Ice fish are scaleless. Their skin is richly supplied with vessels, and a meaningful share of their oxygen uptake happens straight across the body surface rather than at the gills. And there's a piece of this that has to be the most interesting part because it suggests the whole cascade may have been partly automatic. Hemoglobin does a second job that most people never hear about. It mops up nitric oxide. Nitric oxide is a signaling molecule that dilates vessels and drives the growth of new ones. Take hemoglobin out of an animal's blood, and you have removed the main thing that destroys nitric oxide, so nitric oxide levels rise. And the tissues respond by widening vessels and building more of them. Kristen O'Brien and the late Bruce Siddell laid this argument out. And it's a striking idea because it means part of the compensation for losing hemoglobin might have arrived for free, as a chemical consequence of the loss itself. I want to be honest that the field argues about how adaptive any of this really is. There's a strong case made by researchers like Guido Di Prisco and Bruno Toda that losing hemoglobin wasn't an improvement at all. It was a disabling accident that these fish survived only because they were living in the one place on Earth where the environment could absorb the damage, and everything else is expensive patchwork. The word used in that literature is disaptation, which is a wonderfully grumpy way of describing a fish that spent millions of years compensating for a broken gene. Whichever way you read it, the system is real, and it's fragile in a very specific way. Push the temperature up, and the whole arrangement falls over, fast. Work on maximum cardiac performance in these animals has shown that their hearts hit a ceiling at temperatures that a temperate fish would consider a mild spring, because the compensations that let them run on dissolved oxygen only work while demand is being held down by the cold. Warm the water a few degrees, and demand climbs, and there's no reserve to meet it with. A red-blooded fish in that situation just breathes harder and recruits more of its hemoglobin. An ice fish has nothing left to recruit. Some of them went further, which I find almost reckless. In several ice fish species, the heart muscle has also stopped making myoglobin, the oxygen-binding protein inside muscle cells. That loss happened independently at least 4 separate times within the family, in different lineages, from different mutations. The species that lost it tend to have even bigger hearts, as though the animal traded a molecule for more plumbing. So the honest summary of the ice fish is this. It's a vertebrate that solved oxygen delivery by making the whole circulatory system enormous, thin, fast, and cold, and it can only exist inside a narrow band of conditions that happens to occur at the bottom of the world. Could a human body ever do that? Run the numbers, because they're unusually clean. At rest, you consume roughly 250 milliliters of oxygen every minute. Your plasma, without hemoglobin, carries about 0.3 of a milliliter of dissolved oxygen per 100 milliliters of blood at normal atmospheric pressure. Divide the demand by the supply and, assuming you could strip out every last molecule of dissolved oxygen on a single pass, which you can't, you'd need to move somewhere around 80 liters of blood per minute through your heart. Your resting cardiac output is about 5. A well-trained endurance athlete at absolute maximum effort, lungs burning, might reach the mid-30s. Elite rowers and cross-country skiers have been measured a bit higher. So to lie perfectly still on a couch without hemoglobin, you'd need a heart running at roughly twice the peak output ever recorded in a world-class athlete, forever. And you'd need it while your tissues were already starving. At that point, you've stopped describing a heart and started describing a fire pump. There's one way to cheat it, and someone tried. In 1960, the Dutch surgeon Ide Boerema ran an experiment he titled, "With Beautiful Bluntness, Life Without Blood." He took pigs, removed almost all of their red blood cells, replaced the volume with fluid, and put them in a hyperbaric chamber, breathing pure oxygen at several times atmospheric pressure. Cranking the pressure forces far more oxygen into physical solution in the plasma. And the animals lived, consciously, with functioning organs, running entirely on dissolved oxygen, exactly like an ice fish. The catch is the chamber. Boerema needed several atmospheres of pure oxygen to do what the Southern Ocean does for free with cold water. Take the pig out of the tank and it dies within minutes. The ice fish isn't carrying a chamber around. Its chamber is the Antarctic, which is the first version of the pattern I keep noticing in all of this. These animals aren't doing impossible things. They're doing things that are possible only in one place, and they built their entire bodies around the assumption that they'll never leave. Now take that same animal and drop it, not far. A mile, say. Nothing has changed about the temperature, because deep water is cold everywhere. Nothing has changed about the oxygen, at least not yet. But something has arrived that wasn't in the problem before, and it's the insult that I think people understand worst of all. Ask most people what pressure does to a body in the deep sea, and you get some version of crushing, a submarine imploding. The classic image is a foam cup that comes back from the seafloor shrunk to the size of a thimble, which is a real thing that oceanographers do for fun, and which teaches exactly the wrong lesson. The cup shrinks because it's full of air. Air is compressible. You're mostly water, and water barely compresses at all. Take a fish down to the bottom of a trench and its flesh doesn't squash, because there's nothing in the flesh with room to give. Anything with a gas space in it's in immediate trouble, which is why deep-sea fish generally have no swim bladder, and why the ones with swim bladders come up looking turned inside out. Once you remove the gas pockets, the crushing story is basically over. The real damage happens at a scale you can't see at all, and it took physical chemists rather than biologists to explain it. Every chemical reaction in your body and every protein fold involves a change in volume. Not the volume of the protein by itself, but the volume of the protein plus the water shell wrapped around it. When a protein folds, water gets rearranged. When an enzyme binds its target, water gets squeezed out of the contact surface. Some of those steps end up occupying slightly more total space than they started with, and some end up occupying slightly less. Pressure hates the ones that take up more space. If a reaction expands, pressure pushes back against it. If a reaction contracts, pressure encourages it. That isn't a metaphor. It's a hard rule of physical chemistry. And in a cell, it means that under enough pressure, the whole balance of the machinery starts drifting in one direction. Folded proteins begin to loosen because the folded state and the unfolded state differ in volume once you count the water. Enzymes slow down, and not uniformly, so metabolic pathways that were tuned to run in sequence start running at mismatched speeds. Cell membranes stiffen because pressure packs the lipid molecules tighter, and a membrane that was fluid at the surface goes waxy in the deep. And every protein embedded in that membrane, every ion pump and receptor and channel, depends on the fluid around it to move and change shape. Even the internal scaffolding of the cell has a problem. The microtubules that pull chromosomes apart during cell division are held together by an equilibrium that pressure actively pushes apart. Squeeze a dividing cell hard enough, and the spindle just falls to pieces. So the real hazard of the deep sea is a solvent problem. It quietly changes the rules under which every molecule in your body was designed to operate. The numbers get out of hand quickly, Every 33 feet of seawater adds one more atmosphere on top of the one you're already carrying. At the bottom of the deepest trenches, past 26,000 feet, you're looking at something like 800 atmospheres, which works out to roughly 6 tons on every square inch. That's the number people quote. The number that actually matters biologically is that at that pressure, a lot of ordinary proteins from a surface animal simply stop working. Not eventually. Immediately, in a test tube, on the bench. What does it do to a person? We have very good data on this because the offshore diving industry spent decades and a great deal of money finding out, and the results are strange enough that they changed how people think about anesthesia. Below about 100 feet on compressed air, nitrogen starts behaving like a mild drug. That's nitrogen narcosis, the old rapture of the deep, and it feels like a couple of drinks and gets worse with depth, so commercial divers switch to helium, which is far less narcotic. That opens the door to real depth, and then, somewhere around 500 feet and deeper on a helium mix, a different thing shows up that has nothing to do with the gas at all. It's called high-pressure nervous syndrome. Divers start to tremble. Fine tremors in the hands first, then coarser ones, sometimes brief muscle jerks, dizziness, nausea, difficulty sleeping, trouble holding a thought, and characteristic changes in the electrical activity of the brain that show up plainly on an electroencephalogram. It gets worse the faster you compress, and worse the deeper you go. Nobody is fully certain of the mechanism, and I like that the honest answer is still open, but the leading picture is that the pressure itself is directly deforming the behavior of nerve cell membranes and the receptors sitting in them. The same physical effect that unfolds a protein in a test tube is nudging synapses in a person's head, and here's the observation that makes it undeniable. Take an animal under general anesthesia, fully unconscious, and raise the ambient pressure. It wakes up. This is a documented laboratory phenomenon called pressure reversal of anesthesia, and it's been reproduced for decades. Pressure and anesthetics are pulling on the same molecular lever in opposite directions, which means that when a diver at 600 feet feels his hands start to shake, what he is experiencing is, in a real physical sense, the opposite of being put under. The industry pushed on anyway, because there was oil down there. In 1988, on an operation called Hydra 8, a team of COMEX divers worked in the open sea at just over 1,750 feet. Then, in November of 1992, in a steel chamber in Marseille, a diver named Theo Mavrostomos was compressed to 2,300 feet. To get him there, they had to invent a gas. Helium alone would have wrecked him with high-pressure nervous syndrome, so they built a mixture of hydrogen, helium, and oxygen, using hydrogen's mild narcotic effect deliberately as a chemical counterweight to the pressure's effect on his nervous system, fighting one poison with another, essentially, and titrating it by hand. The whole thing took 43 days, 13 days going down. He was at maximum depth for about 2 hours. 23 days coming back up, because the dissolved gas in his tissues had to be released so slowly that rushing it by even a little would have killed him. 43 days of a man's life, an entire specialist gas industry, a pressure chamber built by one of the best engineering outfits in the world. And the number he reached was 2,300 feet. The deepest fish anyone has ever recorded was filmed at 27,349 feet. That fish was a snailfish, filmed in August of 2022 in the Izu Ogasawara Trench south of Japan by a team led by Alan Jameson of the University of Western Australia, working with the Tokyo University of Marine Science and Technology, a juvenile from the genus Pseudoliparus, drifting past a baited lander in total darkness. In the same expedition, they caught 2 of them a little shallower, in the Japan Trench at about 26,300 feet, which are the deepest fish anybody has ever physically had in their hands. If you have never seen one, the appearance is disarming. Snailfish look like a tadpole made of wet tissue paper, Pale, soft, no scales, tiny eyes, a body that's largely gelatinous, a skeleton that's only loosely mineralized. Nothing about them suggests armor, and armor was never the answer anyway. The answer is a molecule called trimethylamine N-oxide. Everyone shortens it to TMAO, and you've met it without knowing. When TMAO breaks down after a fish dies, it becomes trimethylamine, which is the compound responsible for the smell of fish that's been sitting out too long. The smell you associate with seafood going off is the residue of the chemistry that lets seafood exist at depth in the first place. Paul Yancey at Whitman College in Washington State spent a career on this class of compounds. He calls them piezolites. From the Greek for pressure, they're small, cheap, water-soluble organic molecules that an animal dissolves into its tissues in bulk, and they work on the water rather than on the proteins. Here's the mechanism, and it's lovely because it's entirely indirect. TMAO doesn't touch the protein. It actively avoids it. The molecule is unhappy sitting next to the exposed backbone of a peptide chain, so it gets pushed away from protein surfaces into the surrounding water. Now think about what happens when a protein unfolds. Unfolding exposes a lot more backbone, which means more surface that TMAO has to be excluded from, which costs energy, which makes the unfolded state more expensive to occupy. The trick is that nothing ever touches the protein. The water around it has simply been made hostile to the idea of falling apart. That's exactly the right kind of countermeasure for pressure, because pressure is also acting through the water. One is pushing toward the expanded, hydrated, loose configuration. The other is pushing toward the compact, folded one. Load enough TMAO into the tissue, and the two effects cancel across an enormous range of proteins at once, without having to redesign a single one of them. And the evidence for this isn't a nice story. It's a slope. Yancey and colleagues measured TMAO in the muscle of fish and invertebrates hauled from wildly different depths across unrelated groups, and the concentration climbs with depth in a remarkably orderly way. Shallow coastal fish carry a little. Fish from 1,000 meters carry more. Grenadiers from the deep slope carry more again. The relationship holds across species that aren't closely related, which is the signature of physics, driving the outcome rather than shared ancestry. Then they did something braver. They took the slope and extended it downward and asked where it runs out, because there's a ceiling, and osmosis is what sets it. A bony fish in the sea is far less concentrated on the inside than the water around it, something like 1/3 to 1/2 as concentrated, which means the ocean is constantly pulling water out of it. It handles that by drinking and by pumping salt out through its gills, and we'll come back to that whole apparatus shortly. Now start pouring TMAO into its tissues. Every molecule you add raises the internal concentration. Keep adding, and eventually the inside of the fish matches the ocean. Cross that line, and the flow reverses. Water stops leaving and starts coming in, and now the fish has a swelling problem instead of a shrinking one, with none of the machinery to deal with it. Yancey's group calculated where that break-even point falls and published the answer in 2014. About 27,000 feet, roughly 8,200 meters. Below that, they argued, a bony fish shouldn't be able to hold itself together because the only chemistry that keeps its proteins working would have to be loaded past the point where its water balance collapses. The deepest fish ever recorded was filmed at 27,349 feet, 130-odd meters past the prediction, which in this business is a bullseye. Physiology doesn't usually get to make predictions like that and have the ocean cooperate. There are still around 7,000 feet of ocean below that line, in the deepest trenches, and no fish has ever been seen there. Amphipods live down there. Sea cucumbers, anemones, bacteria in real abundance right to the bottom of the Mariana Trench. They can go deeper for a reason that fits the whole argument perfectly. Those invertebrates are already at the same concentration as seawater. They never built a body that runs dilute. So they can load piezolites as high as they like, and nothing osmotic ever goes wrong. The bony fish is locked out of the last stretch of the ocean by a decision its ancestors made hundreds of millions of years earlier, in fresh water, about what a body fluid should be. We have the same molecule in us, by the way, and it does almost nothing. Trimethylamine N-oxide shows up in human blood at low concentrations, and it doesn't come from us. Gut bacteria produce it out of choline and carnitine, which you get from eggs and red meat, and the liver converts the product. Cardiologists have spent the last decade or so arguing about whether elevated levels are a useful marker of cardiovascular risk. That argument is unsettled, and I'm not going to pretend otherwise. But notice the demotion. In a snailfish 5 miles down, this molecule is the load-bearing wall of the entire organism. In you, it's a number on a research panel produced by your bacteria as a side effect of digesting an omelet. And yet we do run the counteraction trick. In exactly one place for a completely different reason. Go into the inner medulla of your kidney, the deep core of the organ where urine gets concentrated. The cells there sit in an environment loaded with urea, because urea is the tool your body uses to build the osmotic gradient that pulls water back out of the tubules. The trouble is that urea is a protein destabilizer. It's literally what biochemists put in a tube when they want to unfold something on purpose. Your kidney cells are living, permanently, in a solution that's trying to take them apart. So they fill themselves with counteracting osmolytes, glycerophosphocylline, betaine, sorbitol, myo-inositol, small organic molecules, accumulated in bulk, doing the same balancing job that TMAO does in a deep-sea fish against the same class of threat. Yancey himself worked on this, and it's the reason the deep-sea story and the kidney story keep turning up in the same papers. You have the chemistry. You have it in a piece of tissue about the size of your thumb, deployed for plumbing. Nobody ever needed it in the muscle of your leg, because nothing in your ancestry ever tried to fold a protein under 800 atmospheres, which is a clean place to leave pressure, because the next insult starts at the surface, and it's one that our lineage did meet, over and over, and lost to almost every time. Some mammals went back to the water. All of them kept the lungs. That's the constraint that everything else in this section falls out of. A fish extracts oxygen continuously from the medium it's swimming in. A seal, a whale, a sea otter, a dolphin, every one of them is carrying a finite tank, and every dive is a countdown. So start with the human version of that countdown, because most people have the physiology of breath holding exactly backwards. You don't run out of air. You run out of patience with carbon dioxide. The urge to breathe is driven overwhelmingly by rising carbon dioxide in your blood, detected by chemoreceptors in your brainstem and in your carotid arteries, and not by falling oxygen. Oxygen has a sensor too, but it's comparatively lazy, and it doesn't shout until things are already bad. So for the first part of a breath hold, you aren't oxygen-starved in the slightest. You're being nagged by a waste gas. Push past the nagging, and your diaphragm starts contracting on its own. Free divers call these involuntary breathing movements, and they're a sort of internal argument between the brainstem and the will. Push past those, and your oxygen keeps falling. And at a certain point, the brain simply stops. There's no warning stage, the lights go out. That is the thing that makes competitive breath holding dangerous in a way it does not look. If you hyperventilate first, you blow off carbon dioxide and delay the urge to breathe, which sounds helpful and is the opposite of helpful, because you have removed the alarm without adding any oxygen. Divers black out underwater with no idea it's coming. Every serious freediving organization builds its entire safety structure around that fact, with a trained buddy on the surface, one diver in the water at a time, and no exceptions. I mention it because people watch these videos and go to the pool, And this is the one part of this script that could actually hurt someone. Do not practice breath holding in water alone. Ever. The competitive numbers for calibration. An untrained adult sitting still manages something like 30 seconds to a minute before the urge to breathe becomes unbearable. Trained static apnea, lying motionless in a pool on ordinary air, has reached a bit over 11 minutes. There are longer records, well over 20 minutes, but those involve breathing pure oxygen beforehand, which loads the blood and lungs far past what air can do, and they're a different event. 11 minutes, lying perfectly still, in warm water, at the surface, after years of training. That's the human sealing. A Weddell seal routinely goes for 20 minutes while hunting, at depth, in the dark, in freezing water, doing physical work. Its recorded maximum runs past an hour. It takes 4 separate systems stacked on top of each other, and every one of them exists in your body in a form so faint that most people have never noticed it operating. The first is the reflex itself. Put your face in cold water and hold your breath, and your heart rate drops. This is real. You can measure it with a fingertip monitor, and it happens because cold receptors around your nose and forehead feed into the trigeminal nerve, which talks to the part of the brainstem that runs your heart. At the same time, the vessels in your fingers and toes narrow. Physiologists have known about this since Per Scholander's work in the middle of the last century, and he called it the master switch of life, which is the kind of phrase you can only get away with if you're also right. In you, the switch throws a little. Heart rate falls maybe 10 to 30%. Trained apneists get more out of it, sometimes a lot more, but the effect is modest and it doesn't last. In a Weddell seal, the same switch takes a resting heart rate of around 60 or 70 beats a minute, down into the single digits. 4 beats a minute has been recorded. 4. And the heart is doing it on purpose. Holding a rhythm that would be a medical emergency in any land mammal for 20 minutes at a stretch, and then climbing straight back to normal on surfacing. The second piece is where the blood goes. And this is the part that reorganizes the animal into something functionally different. The arteries feeding the muscle, the gut, the kidneys, and the skin clamp down so hard that those tissues are effectively taken off the circulation. Kidney filtration in a diving seal basically stops. The blood that remains in play is servicing the heart and the brain, which can't tolerate any interruption. And in a pregnant female, the placenta. The muscles, meanwhile, are still working. They just work without a blood supply, burning through their local stores anaerobically and filling up with lactate. And because the vessels are shut, that lactate has nowhere to go. It stays penned in the muscle for the whole dive. Then the seal surfaces, the vessels open, and the whole load hits the bloodstream at once. Scholander noticed this in the 1940s. Blood lactate in a diving seal spikes after the dive is over, which sounds backwards until you realize it's the fingerprint of a limb that was sealed off from the rest of the animal. Try that in a person, and you've described a compartment syndrome or a tourniquet injury. The seal does it on every hunting trip and swims home. The third piece is the tank size, and it comes in 2 parts. A human adult carries roughly 70 milliliters of blood per kilogram of body weight. Somewhere around 7% of you, give or take. A deep diving seal runs 2 to 3 times that, up past 200 milliliters per kilogram, so a fifth or more of the animal's mass is blood. It's a body built around a reservoir, and that blood is thicker with red cells. A healthy human hematocrit sits in the low 40s, and doctors get nervous when it climbs much past 50 because thick blood clots. Diving seals run in the 60s, and they get away with it partly because so much of the load isn't in circulation most of the time. And that leads straight to the organ I think is the most underrated thing in this entire video: the spleen. Your spleen is a filter and an immune organ. It holds a couple hundred milliliters of blood, and when you exercise hard or hold your breath, it squeezes some of that out into circulation. The effect is measurable and small, a few percentage points of hematocrit. A seal's spleen is a warehouse. Relative to body size, it's many times larger than yours. It's packed with red cells, and it's under direct control of the nervous system through catecholamines. Work in the 1990s on free-diving Weddell seals showed the pattern plainly. Hemoglobin concentration measured right after a dive was substantially higher than during quiet rest, and hematocrit jumped from the mid-40s into the mid-50s within the first couple of minutes after surfacing. Researchers estimated that a large Weddell seal keeps something on the order of 20 liters of packed red cells parked outside the main circulation and injects them when it dives. 20 liters. That's more total blood than a whole adult human contains, held in reserve, as a second fuel tank. Elephant seals go further and add a second reservoir, an enormous swollen section of the vena cava sitting near the liver, a hepatic sinus that acts as another storage tank. Northern elephant seal pups, studied under controlled dives, showed both the spleen and that sinus loading and unloading as part of the same coordinated response. And here's where the human comparison stops being purely humiliating, because there's one population that started building this. The Bajau are a group of maritime peoples living across Indonesia, Malaysia, and the Philippines who have been diving for their food, on a single breath, for something like 1,000 years. Not recreationally, as a job most days, for generations. In 2018, a team led by Melissa Ilardo published a study in the journal Cell comparing Bajau individuals with a neighboring farming population, the Saluan, who live in the same region and share a lot of ancestry but don't dive. They imaged spleens with portable ultrasound. The Bajau spleens were about 50% larger, and it wasn't a training effect because the difference showed up in Bajau individuals who don't dive at all. The team found signals of natural selection around a gene called PDE10A, which is involved in thyroid hormone signaling. And thyroid hormone is known to influence spleen size in mammals. 1,000 years of holding your breath for a living, and the human genome has begun, tentatively, to build the seal's second fuel tank. That's a very small step compared with what a Weddell seal is carrying. And it isn't nothing. And I think it's the single most important data point in this entire subject. We aren't built differently in principle. We're just not old enough at this. The fourth piece of the diving system is the one I've been saving, because the mechanism behind it's quietly the cleverest thing in this whole subject. Blood is only half of an oxygen store. The other half sits inside the muscle itself, in a protein called myoglobin. Myoglobin is hemoglobin's smaller cousin, one iron-holding unit instead of 4, and its job is to hold oxygen inside the muscle cell so the cell has a local supply when the blood supply is interrupted. It's the reason a steak is red and a chicken breast is white. Human skeletal muscle carries roughly 4 to 8 milligrams of myoglobin per gram of tissue. Values vary by muscle group and by training, but that's the neighborhood. Deep diving seals carry somewhere between 50 and 100 milligrams per gram, an order of magnitude more. Whale muscle in the same range or higher. If you have ever seen a photograph of the inside of a deep diving whale, the muscle isn't red, it's nearly black, and the darkness is the myoglobin. That tissue is a battery. Now, why does that not simply work for us? Why can't a person train up to seal levels? Because there's a hard sealing and it's a solubility problem. Proteins packed together at very high concentration stick to each other. They aggregate. They come out of solution. They form clumps. And clumped protein in a muscle cell is a disease. Half the neurodegenerative conditions we know about are protein aggregation stories. Pour myoglobin into a cell at 10 times the human concentration and ordinary myoglobin should precipitate. In 2013, Scott Merceta and Michael Barenbrink and their colleagues at Liverpool published the answer in Science, and it's beautifully simple. They compared the myoglobin sequences of a large number of mammals and calculated the net electrical charge on the outside surface of the molecule. In diving mammals, that surface charge is higher, consistently. And it went up independently in all 8 separate lineages of mammals that took to the water, seals, whales, otters, manatees, the lot, using different amino acid substitutions each time to reach the same electrical result. Like charges repel, load a cell with myoglobin molecules that all carry the same surplus charge on their surfaces, and they push each other apart. They physically can't stack up and clump because every molecule is shoving its neighbors away. The animal can then concentrate the protein to a level that would be chemically impossible with the standard version. They did one more thing with that finding, and it's the kind of result that makes you sit back. Because surface charge is readable straight off a gene sequence, and because gene sequences can be reconstructed backward along an evolutionary tree, the team estimated the myoglobin charge of extinct ancestors, and from that inferred roughly how long those extinct animals could hold their breath. They read the diving ability of a creature nobody has ever seen off the electrostatics of a protein that no longer exists. Your myoglobin doesn't have that surface. Nothing in the last 200 million years of our ancestry ever needed it. There's one more component, and it's the piece that solves a problem the other 3 would otherwise create: depth. If you hold a full breath of air and go down, the gas in your lungs is under increasing pressure, and pressurized gas dissolves into your blood. That's where decompression sickness comes from. Come up too fast, and dissolved nitrogen comes back out of solution as bubbles inside your tissues. Scuba divers manage it with tables and slow ascents. Breath hold divers were assumed to be immune because how much nitrogen can one lungful contain? Not immune, as it turns out. Pearl divers in the Tuamotu archipelago of French Polynesia have long described a condition they call terravana, with vertigo, paralysis, and worse, appearing after long days of repeated deep dives. The current understanding is that it's decompression sickness, accumulated one breath at a time across dozens of descents. Seals and whales solved this by letting their lungs collapse on purpose. Somewhere between about 100 and 160 feet down, the air spaces in a diving seal's lungs give way. The alveoli, the tiny bubbles where gas exchange actually happens, flatten out. The air inside them gets pushed up into the conducting airways, the windpipe and the bronchi, which in these animals are unusually stiff, reinforced with cartilage all the way out toward the small branches, specifically so they won't collapse when everything else does. And once the air is sitting in a rigid pipe with no blood supply around it, gas exchange stops completely. Which means that for the entire deep portion of the dive, no nitrogen is entering the animal's blood at all. The gas is sealed in a part of the respiratory tract that can't deliver it anywhere. They stack a second precaution on top of that. Many seals dive after breathing out, not in. They go down deliberately underinflated, which cuts the gas load further and makes them less buoyant on the way down, so they spend less energy descending. Your chest does something related, and it's the reason competitive freediving is possible at all. As you descend, the gas in your lungs compresses, and your lungs would be pulled into a vacuum inside a rigid ribcage. Instead, blood shifts into the vessels of your chest and fills the space. Physiologists call it blood shift, and it's unmistakably a marine mammal trait showing up in a human. It's the reason Herbert Nitsch could ride a weighted sled to about 700 feet in 2007 and come back up with a chest. But ours is a partial version. Freedivers still suffer lung squeeze, Brigabris Windries toe squeeze, bleeding into the airways when the shift can't keep up with the descent. And it happens at depths a seal treats as the shallow end. I do want to say plainly that marine mammals aren't magically exempt from any of this. Beaked whales that stranded during and after naval sonar exercises have been examined by pathologists who found gas and fat bubble lesions in their tissues, consistent with something that looks a great deal like decompression sickness. The leading interpretation, argued in Nature by Paul Jepson's group and developed by others since, is that disturbance changes the animal's diving behavior in a way their physiology was never built to handle. The system is exquisite. It's also tuned to a specific set of conditions, and it can be pushed outside them, which makes what those animals do when nobody is bothering them even more remarkable. Cuvier's beaked whale isn't an animal most people could name. Gray, blunt-headed, scarred, roughly the size of a small bus, spends almost all of its life out of sight. In 2014, satellite tags recorded one diving to 9,816 feet off the coast of California, nearly 2 miles straight down, on one breath. Then, in 2020, a team led by Nicola Quick at Duke University, tagging these whales off Cape Hatteras in North Carolina, recorded a single dive that lasted 3 hours and 42 minutes. The previous record had been about 2 hours and change. This beat it by more than an hour. And the physiology doesn't fully explain that. Researchers calculate something called the aerobic dive limit, the point beyond which an animal must be running on anaerobic metabolism, based on its oxygen stores and its metabolic rate. For Cuvier's beaked whale, the estimate sits somewhere around half an hour. The record dive was 7 times that. The team's own paper is careful about it, noting that the longest dives may be unusual events, possibly responses to disturbance, and that we don't have a complete account of how the animal pays for them. I find that reassuring rather than frustrating. The deepest diving mammal on the planet is doing something that our best model of mammalian diving can't yet fully cover, and the honest people studying it say so. Step back and look at what the diving system actually is, though, because it's the clearest example in this whole subject of an animal facing several insults at once. A beaked whale on a deep dive is dealing with oxygen shortage, hydrostatic pressure, and cold simultaneously on a schedule, with no option to abort. And the solution wasn't one clever organ. It was a heart rate that drops to almost nothing, a circulatory system that reroutes itself mid-dive, a spleen the size of a warehouse, a blood volume triple hours, a muscle protein rebuilt at the level of surface electrostatics so it can be packed to concentrations that would otherwise turn the muscle into sludge, and a respiratory tract engineered to shut itself down on the way down. 6 systems, coordinated, none of which we have in any meaningful form. There's one insult, though, that even the whales never solved. They dodged it. A whale doesn't drink. Neither does a seal, for the most part. They get their water from their food and from the metabolic water released when they burn fat. And they run kidneys, efficient enough to make that work. The ocean is a desert to them, chemically speaking, and their answer was to never take a sip of it. The fish didn't get that option. Salt is the insult people take least seriously, and it's the one that would kill you fastest if you were dropped in the ocean with a life jacket and nothing else. The physics is embarrassingly simple. Put a membrane between 2 solutions of different concentration, and if water can cross that membrane but the dissolved material can't, water will move toward the saltier side. Every cell in your body is wrapped in exactly that kind of membrane, studded with dedicated water channels called aquaporins that make it even more permeable than it would otherwise be. Your cells cannot refuse. There is no valve. Water goes where the chemistry sends it. Your blood plasma runs at roughly 290 millismoles per liter, which is the unit for counting dissolved particles. Seawater runs around 1,000 to 1,200, 3 to 4 times as concentrated. So a castaway drinking seawater is loading their bloodstream with salt that has to be excreted. And the only organ that can excrete, it's a kidney with a hard ceiling. The human kidney can concentrate urine to about 1,200, maybe 1,400 millismoles per liter. And that's it. That number is set by the architecture of the loop of Henle and the countercurrent gradient in the medulla. and no amount of thirst or willpower moves it. Now do the accounting. Drink a liter of seawater, and you've taken on roughly the same quantity of dissolved solute that a liter of your maximally concentrated urine can carry away. Break even at best. Except you also have your own day's worth of waste to get rid of. Urea and acid and potassium and the rest. Several hundred millimoles of obligatory solute that need water to leave in. So you finish the transaction down on water, not up. Drinking seawater makes you thirstier. Doing it repeatedly is how people die faster than they would have if they had drunk nothing at all. There's a bonus insult on top. Seawater carries magnesium and sulfate, and those two, taken orally in quantity, do to your gut roughly what a laxative does, which costs you more water still. Meanwhile, sitting on the water above you, an albatross is drinking the ocean and doing fine. Seabirds have a salt gland, usually above the eye, that produces a fluid far saltier than seawater and drips it out through the nostrils. Sea turtles have one that empties near the eye, which is where the myth of the weeping turtle comes from. The marine iguanas of the Galapagos sneeze theirs out in visible white crusts, which is why they look like they've been in a flower fight. Sharks have one too in the rectum. Mammals never built the gland, and it isn't that mammal kidneys are incapable of impressive things. A kangaroo rat concentrates its urine past 5,000 milliosmoles, 4 times what you can manage, and lives its entire life without drinking water. The equipment exists in our class of animal. Our ancestors just never spent long enough somewhere that would have selected for it. Marine fish had no such luxury, because they're wet on both sides. A bony fish in the sea has the same problem the castaway has, permanently, through its entire skin and gill surface. Its body fluids run at roughly 1/3 to 1/2 of seawater concentration, an inheritance from freshwater ancestors hundreds of millions of years back. And the ocean is pulling water out of it every second through the enormous, thin, highly permeable surface of the gills. The gills have to be thin and enormous. That's how gills work. The organ that keeps the fish breathing is also the leak. So marine fish drink constantly. A saltwater fish takes seawater in through its mouth deliberately, all day, which is the exact behavior that would kill you. Then it does the hard part in 3 separate places. In the gut, it absorbs the water along with sodium and chloride, which sounds self-defeating until you realize the salt is the thing that drags the water across the intestinal wall in the first place. The magnesium and sulfate mostly stay behind and get dumped. The kidney handles the divalent ions and produces a tiny trickle of urine because water is the scarce resource and you don't throw scarce resources away. And then the sodium and chloride that came in with the water have to go back out against a steep concentration gradient into water that's already saltier than the fish. That's uphill transport, and it happens in specialized cells in the gill called ionocytes. The mechanism is a 3-part relay, and it's worth walking through slowly, because once you see it, you understand why we could never bolt it on. On the inner face of the ionocyte, facing the blood, sits an enzyme called sodium-potassium ATPase. It burns cellular energy to push sodium out of the cell into the blood, which leaves the inside of the cell low in sodium. That gradient is the power source for everything that follows. Also on the interface is a cotransporter, NKCC, which uses the sodium rushing back down its gradient into the cell to drag chloride in alongside it. Chloride now builds up inside the ionocyte, well above where it would sit on its own. And on the outer face, the side touching the ocean, sits a chloride channel. It's the same protein family as CFTR, the one that's broken in cystic fibrosis in humans. Open that channel, and the accumulated chloride pours out into the sea. Sodium follows through leaky gaps between neighboring cells. Pulled by the electrical charge the chloride left behind. Energy in, salt out, against the gradient, continuously, for the animal's whole life. None of that's free. Sodium-potassium ATPase is one of the great energy sinks in animal biology, and a fish running its gills as a salt pump is spending a real slice of its metabolism on staying the right concentration. Estimates of how big that slice is vary wildly across studies, from a few percent up to a quarter or more depending on the species and the method, and the honest version is that nobody has a single clean number. What everyone agrees on is that it's one of the standing costs of being a fish in the sea, paid every hour, forever, whether the animal eats or not. Now flip the direction, because the second half of this problem is where it gets properly strange. Take that same fish and put it in a river. Everything reverses. The water outside is now more dilute than the fish, so water floods in instead of leaking out, and salts leak out instead of flooding in. A freshwater fish is bailing. It stops drinking entirely because the last thing it needs is more water, and it produces urine in enormous dilute volumes, in some species approaching a third of its body weight in a day. And its gills, instead of pumping salt out, now have to scavenge sodium and chloride from water that has almost none, using a different set of pumps, a proton pump, and an exchange system that trades bicarbonate for chloride. Same organ, opposite job. Different molecular hardware. Before we get to the animals that can do both, it's worth noticing that a human being sits in a narrow window in exactly the same way, and we fail off both edges. Too much salt and not enough water, which is the castaway, gives you hypernatremia. Cells shrink, and the ones that mind most are in the brain. Too much plain water and not enough salt gives you the opposite. Exercise-associated hyponatremia turns up most famously in marathon runners who drank diligently at every aid station and diluted their blood sodium enough that water started moving into their brain cells. Brains swell inside a rigid skull. There are documented deaths from this in healthy athletes at well-organized races, and the guidance that came out of it was to drink to thirst rather than on a schedule. Our tolerance for the concentration of our own blood is something like plus or minus a few percent. That's the entire range. A killifish laughs at that. Fundulus heteroclitus is a small, undistinguished, extremely tough fish that lives in salt marshes along the Atlantic coast of the United States. In a marsh, the salinity swings depending on the tide, the rain, and the time of day. That animal can move from fresh water to water several times saltier than the open ocean. And it doesn't just tolerate the change, it rebuilds its gills. The ionocytes remodel, the transporters get redistributed. The whole apparatus reconfigures over hours to days, and then it reconfigures back. The version of this that gets the most attention, and deserves it, is what a salmon does. An Atlantic salmon hatches in fresh water and lives there as a par for a year or more, running the freshwater program, scavenging ions in, urinating a river's worth. Then, in spring, something trips. And what trips its day length? Lengthening days, sensed as photoperiod, in a fish that has passed a size threshold, set off the par-smolt transformation. The animal changes color, changes shape, changes behavior, starts swimming downstream. And inside the gills, the ionocytes switch which version of the sodium pump they're building. There are different isoforms of the sodium-potassium ATPase alpha subunit, one associated with freshwater ion uptake and another associated with seawater ion secretion, and the fish shifts production from one to the other. The seawater machinery ramps up while the fish is still in the river. It retools before the insult arrives. On a calendar cue, that's a fish preparing for an environment it has never encountered, based on day length, weeks in advance. And then, years later, it comes back up the river and reverses the entire thing. There's a whole separate solution to the salt problem, and the sharks and rays took it, and it connects straight back to the deep sea. A shark doesn't fight the ocean's concentration. It matches it. Shark blood is loaded with urea, the same nitrogen waste product your kidneys work to get rid of, retained on purpose at concentrations that would put a person in a coma. We're talking about something on the order of 50 times the level a human physician considers normal. On top of the urea, the shark stays slightly more concentrated than seawater, so water trickles gently inward rather than being dragged out. And the animal gets rid of the excess through its kidney and secretes leftover salt through that rectal gland. Urea at those concentrations should shred the shark's proteins. Urea is the classic laboratory denaturant. So the shark carries a counterweight, and you already know what it is. TMAO, the same molecule the snailfish uses at 8,000 meters. Sharks carry urea and TMAO together at a ratio of roughly 2 to 1, and at that ratio, the destabilizing effect of the urea and the stabilizing effect of the methylamine very nearly cancel out. It's a chemical balancing act running in the blood of every shark in the ocean. And when Yancey's group looked at deep water sharks and rays, they found the ratio shifts with depth. The deeper the animal, the more TMAO relative to urea, because now the molecule is doing two jobs at once, canceling the urea and canceling the pressure. Same compound, two entirely different insults, one solution. The interesting exception, since somebody is going to bring it up, is the bull shark, which swims up rivers. People have caught them in the Mississippi, as far north as Illinois, and there's a resident population in Lake Nicaragua. When a bull shark moves into fresh water, it dials the urea retention down because now it's the one that's too concentrated and the river is trying to flood it. It runs both programs. Most sharks cannot. Everything in this section, every pump, every gradient, every gram of urea, has one thing in common. It runs on energy. And energy in an animal means burning fuel with oxygen. So what happens when there's no oxygen? Not a little less, almost none, because there are places in the open ocean, enormous places, where that's the standing condition. And they aren't exotic corners. They sit right off the coast of Mexico, off Peru, in the Arabian Sea, off Namibia. And they're some of the most biologically productive waters on the planet. They're called oxygen minimum zones, and the way they form is a little grim and entirely logical. Sunlit surface water over an upwelling zone is packed with nutrients, so plankton grow like a lawn in June. Everything that lives up there eventually dies and sinks. On the way down, bacteria consume that falling material, and consuming it uses oxygen. If the deeper water in that region is also poorly circulated, so nothing is bringing fresh oxygen in from the poles, then the bacteria simply strip the layer bare and nothing refills it. The result is a band, usually somewhere between about 600 and 3,000 feet, where oxygen falls off a cliff. Surface seawater carries a couple hundred micromoles of oxygen per kilogram. In the core of a strong oxygen minimum zone, it can drop below 20. And in the worst of them, below 5. That's a couple of % of what the surface holds, and in the very center, it's functionally nothing. To calibrate what that means for an air-breathing animal, we have unusually good numbers, because a group of doctors carried a blood gas kit up Mount Everest. The Caldwell Extreme Everest Expedition, in 2007, had climbers draw arterial blood at 27,559 feet on the way down from the summit. Nobody had ever done that. The results were published in the New England Journal of Medicine, and they're still startling. At sea level, the oxygen pressure in your arteries sits somewhere around 95 to 100 millimeters of mercury, and your hemoglobin is about 97% saturated. In those 4 climbers high on the mountain, the average arterial oxygen pressure was about 25. The lowest single value was 19. For context, if you walked into an emergency room with an arterial oxygen pressure of 19, you'd be intubated before you finished your sentence. These men were climbing. So what did their bodies do to get there? Everything we have, which is a short list. They hyperventilated massively. which raises the oxygen pressure in the lungs at the cost of blowing out carbon dioxide and turning the blood alkaline. Their hearts sped up. Over weeks of acclimatization, they made more red blood cells, driven by erythropoietin from the kidney sensing the shortage. Capillary density in muscle shifts. Mitochondrial content actually goes down, which sounds backwards and appears to be a real remodeling response. Notice what isn't on that list. At no point does a human being change the oxygen affinity of their hemoglobin. The binding curve of the molecule is essentially fixed. There's a bit of tuning available through a compound called 2,3-bisphosphoglycerate, which the red cell makes more of at altitude and which loosens hemoglobin's grip on oxygen. That helps release oxygen to tissue and hurts your ability to pick it up in the lungs. Meanwhile, the alkaline blood from all that hyperventilating tightens the grip, which helps you load and hurts you unload. Your body is pulling the same lever in both directions at once and roughly canceling itself out. That's what you get when two emergency responses that were never meant to run together fire at the same time. Other vertebrates did the obvious thing instead. The bar-headed goose flies over the Himalayas twice a year, and it carries a hemoglobin with a small number of amino acid changes that give it a distinctly higher affinity for oxygen than its lowland relatives have. One substitution in particular loosens a contact inside the molecule and shifts the whole curve. The goose loads oxygen at altitudes where a duck's blood would come up half empty. Deer mice living high in the Rockies have their own version of the same solution. Humans living at altitude for thousands of years didn't get that. Highland Tibetans, who have been up there longer than almost anyone, went in a completely different direction, carrying lower hemoglobin concentrations than lowlanders do at altitude, along with variants around oxygen-sensing genes and unusually high circulating nitric oxide that keeps their vessels open and blood flowing. They optimize delivery and flow rather than the binding chemistry itself. And there's one more thing we can't do at all, which matters more than any of it. We can't turn ourselves down. A mammal's metabolic rate is close to non-negotiable. You're warm-blooded, which means a large part of your energy budget is spent producing heat you immediately lose. And you can't switch that off because your enzymes only work in a narrow temperature band. Your brain alone burns something like a fifth of your resting energy, and it doesn't accept a reduced allowance. Hibernators have a version of the off switch. We do not. The one crude method medicine has for lowering a human's oxygen demand is cooling. Targeted temperature management after cardiac arrest, cooling the body down a few degrees to buy the brain some margin while the heart is being sorted out. That's our whole toolkit for demand reduction, and it requires a hospital. An animal living in an oxygen minimum zone treats that as a routine Tuesday. Consider the Humboldt squid, Dosidicus gigas. It's a large, aggressive, extremely fast predator off the Pacific coast of the Americas, and it has one of the highest metabolic rates ever measured in an animal. Squid swim by jet propulsion, which is a spectacularly inefficient way to move, and they pay for it in oxygen. That animal spends its days inside the oxygen minimum zone. Every night it rises into the productive surface layer to hunt, and every dawn it descends back into a layer of water with almost no dissolved oxygen in it, and it stays there for hours. Brad Seibel and colleagues measured what happens during those hours, and the answer is that the squid turns itself down. Under sustained low oxygen, its metabolic rate drops dramatically, by most of its normal value, and it sits in a state of suppressed activity waiting for night. But suppression alone wouldn't be enough, because the squid still has to extract oxygen from water that barely has any. That takes a different piece of hardware. Cephalopods don't use hemoglobin. Their oxygen carrier is hemocyanin, which uses copper instead of iron, floats free in the plasma rather than being packaged in cells, and turns blue rather than red when it's carrying oxygen. Squid blood is blue. That fact usually gets filed under trivia, and it's actually the doorway into the whole solution. Because a dissolved carrier can be tuned in ways a packaged one can't, and the Humboldt squid's hemocyanin is tuned to an extraordinary degree by two things that change as the animal moves up and down: temperature and acidity. Down in the cold, dark, oxygen-poor layer, the hemocyanin's affinity for oxygen is high. It grabs what little oxygen is there and holds it. Then the squid rises at night into warmer water and starts hunting. And hunting means burning fuel. And burning fuel produces carbon dioxide, which makes the blood more acidic. Both the warmth and the acidity slam the affinity down. The molecule lets go of its oxygen, right where the muscle needs it. The sensitivity to pH in this animal is unusually strong even for a cephalopod. Seibel's measurements describe a molecule whose oxygen binding shifts steeply with a small change in blood acidity, which is exactly what you want if you're using acidity as your delivery trigger. So the squid is carrying a gearbox, and the vertical migration itself throws the gears. There's a cost buried in that, and it's worth stating because it's a real vulnerability rather than a poetic one. An animal whose oxygen delivery depends this heavily on blood pH is exposed to anything that changes ocean chemistry. Work by Rui Rosa and Brad Seibel, published in the Proceedings of the National Academy of Sciences, exposed Humboldt squid to water with elevated carbon dioxide, of the kind projected for the coming century, and found their metabolic rates and activity dropped substantially. The molecule that makes them formidable is the molecule that makes them fragile. And then there's the animal that took the opposite strategy to its logical extreme. Vampiroteuthis infernalis, vampire squid from hell, which is a real Latin name and one of the great acts of scientific melodrama. It's a small, dark red, gelatinous cephalopod with enormous eyes and webbing between its arms. And it's neither a squid nor a vampire, being the last surviving member of its own order. It lives inside the core of the oxygen minimum zone, permanently, not passing through, resident. Its hemocyanin has the highest oxygen affinity ever measured in any cephalopod, where the Humboldt squid's carrier is tuned to release oxygen aggressively. The vampire squid's is tuned to grip it, extracting oxygen from water that would leave most animals suffocating. And its metabolic rate is the lowest measured in any cephalopod, by a wide margin. It achieves that partly by being made largely of water and jelly, with very little muscle, and by carrying ammonium ions in its tissues to make itself neutrally buoyant, so it doesn't have to swim to stay in place. It hangs there. Henk Jan Hoving and Bruce Robison, using remotely operated vehicles at the Monterey Bay Aquarium Research Institute, worked out what it eats. And the answer settles the whole argument. It does not hunt. It unrolls a long, sticky, retractable filament, drifts, and collects marine snow. The endless slow fall of dead plankton and mucus and debris coming down from the sunlit water above. Then it scrapes the filament clean and eats what it caught. It solved the oxygen problem by needing almost none, and it bought that by giving up muscle, speed, and predation entirely. That's the trade, stated plainly, and it's been paying off for a very long time. The pattern in that layer of the ocean repeats across unrelated groups, which is the giveaway that the water is doing the designing. Fish that specialize in oxygen minimum zones tend to have oversized gills for their body mass, hemoglobins that bind oxygen unusually tightly, and metabolic rates well below what you'd predict from their size. Crustaceans down there show the same tightening of the binding curve. Nobody coordinated it. The physics of a low oxygen layer produces the same shopping list every time. And there's an ecological twist that I didn't expect the first time I ran into it. The oxygen minimum zone isn't only a hazard, it's a hiding place. If you can tolerate 2% oxygen and your predators can't, then that layer is the safest real estate in the ocean. A great deal of the daily vertical migration in the sea, the largest mass movement of animals on the planet, which happens every single night, is animals commuting between food at the top and safety below. Some of that safety is darkness. Some of it, in these regions, is chemistry that other animals can't handle, which is a good moment to mention that these zones are getting bigger. A large analysis published in Nature in 2017, led by Sönke Schmidtko, put the global loss of dissolved oxygen from the ocean at around 2% since 1960, with the low oxygen regions expanding faster than that. Warm water holds less gas, and a more stratified ocean mixes less. The measurement is solid. Exactly what it does to the animals that live there over decades isn't something anyone can state with confidence yet, and I'd be suspicious of anyone who told you otherwise. Now follow one of those migrating animals upward, all the way up, past the depth where the squid stops, into the top of the water column where the light is. Because there's an insult up there too, and it's the one we recognize the most and understand the least. Sunlight isn't a single thing. The visible part is a narrow band, and just past the violet edge sits ultraviolet, which carries enough energy per photon to break chemical bonds. Ultraviolet A is the long wavelength portion, and it penetrates deepest, into skin and into water. Ultraviolet B is shorter, more energetic, and more directly destructive. Ultraviolet C would be worse than either, and the atmosphere eats essentially all of it before it arrives, which is the only reason this conversation is possible. What ultraviolet B does to a cell is specific and it's chemical. It's absorbed directly by DNA, in particular by adjacent pairs of the letter T sitting next to each other in the sequence, and the energy fuses them into a joined structure called a thymine dimer. Now the strand has a kink in it. The enzymes that copy DNA hit that kink and stall or misread it, and if the repair machinery doesn't get there first and cut the damage out, a mistake gets written permanently into the code. Your cells do this repair constantly. There's a whole crew of enzymes, nucleotide excision repair, dedicated to spotting these lesions and excising them. People born with a fault in that repair system have a condition called xeroderma pigmentosum, and they have to avoid daylight almost entirely, which is a stark illustration of how much damage is being quietly cleaned up in the rest of us every sunny afternoon. Ultraviolet A works differently, mostly indirectly, generating reactive oxygen species that damage DNA, proteins, and membranes by chemical collateral. And the human defense, the one we all know, has an interesting property that turns out to be the whole reason marine animals couldn't use it. Melanin is a pigment. It sits in packages in your skin cells, positioned over the nucleus like a parasol, and it absorbs ultraviolet before it reaches the DNA, dissipating the energy as heat. It's extremely good at its job. Populations with more of it have dramatically lower rates of skin cancer, and the geographic gradient of human skin color tracks ultraviolet intensity closely enough that it's one of the cleanest examples of natural selection anyone has on our species. The catch is that melanin is dark, and the darkness is the mechanism itself. Absorbing light across a broad range is exactly what makes a substance look black or brown. Now imagine you're a coral. Inside your tissues live symbiotic algae that you depend on for most of your food, and they need sunlight to photosynthesize. Turning your surface dark to block ultraviolet would starve them. Imagine you're the lens of a fish's eye. Your entire function is to be transparent. Imagine you're a copepod, or a fish egg, or a plankton drifting in the top few feet of a tropical sea. You're transparent because transparency is your only defense against being seen and eaten. Going dark means being spotted. The ocean is full of organisms for which the human solution isn't available at any price, and so it evolved something else. And this one is my pick for the most underrated molecule in the sea. They're called mycosporin-like amino acids, which is a terrible name for a wonderful thing, and everyone in the field shortens it to MAAs. They are tiny, under 400 daltons, which in molecular terms is a pebble. They dissolve in water. And crucially, they're colorless to the eye because their absorption sits entirely in the ultraviolet, in a band roughly from 310 to 360 nanometers, which covers most of ultraviolet B and a good chunk of ultraviolet A. Chemically, they're built around a ring, a cyclohexanone or cyclohexenamine, with amino acid pieces hung off it. When an ultraviolet photon hits, the molecule absorbs it. The energy gets shuffled internally, and it comes back out as a small amount of heat. The molecule itself is unchanged and does it again. No free radicals produced, no damage to the compound, no breakdown product to dispose of. Just an extremely efficient, colorless, reusable photon sponge. The family has members with names that read like a spell. Chinarene, Porphyra 334, Palaethine, named for the zoanthid genus Palaethoa, where it was found, Asterina 330, Palaethanol, Palaethine, Mycosporin glycine. Each one peaks at a slightly different wavelength, and organisms carry combinations, which effectively broadens the coverage. They're everywhere in the sea. A survey of Antarctic marine organisms found them in the overwhelming majority of species examined, something close to 9 in 10. Corals are full of them. Sea urchin eggs, sponges, fish, algae, cyanobacteria. There's a version of this in the mucus that coats a reef fish, meaning the animal is wearing a chemical film that filters ultraviolet. And now the detail that makes this the sharpest comparison in the whole video. Animals cannot make them. The synthesis route runs through the shikimate pathway, a piece of biochemistry that bacteria, fungi, algae, and plants have, and that animals, as a group, don't. Which means a coral, a fish, a sea urchin is getting its sunscreen from somewhere else entirely, from its symbiotic algae if it has any, or straight out of its diet if it doesn't, and then transporting it and depositing it in the tissues that need protecting. So a marine animal has a system for absorbing a specific compound out of its food and installing it in its skin and its eyes as a filter. Call that what it is, a system with uptake, transport, and targeted deposition, existing for one purpose. The experiment that nails it's about as blunt a piece of study design as biology gets, and I love it for that. Researchers fed Medica, a small Japanese freshwater fish, a diet containing these compounds, and fed the same compounds to hairless laboratory mice. Then they went looking for the molecules in the tissues. The fish accumulated them. The mice did not. Same food, same molecules going in. The fish has the transport machinery to pull them out of the gut and put them where they'll do some good, and the mammal simply doesn't. We eat seaweed. We eat shellfish that are loaded with these things. It passes straight through us. Where the fish puts them is the detail that ties it back to the melanin problem. A lot of it goes to the eye. The cornea and the lens, tissues that must stay perfectly clear and that sit directly in the path of every photon coming down through the water, get loaded with a transparent ultraviolet filter. You can't solve that problem with a pigment. There's no dark lens. Corals get theirs from the symbiotic dinoflagellates living in their tissues, the same algae that feed them, and shallow water colonies generally carry more than deep ones, which is exactly the direction you'd predict. There's an uncomfortable implication in that arrangement, since a coral that expels its algae under heat stress is also expelling its sunscreen supply at the moment when it's sitting in warm, bright, shallow water. Researchers have raised that as a compounding factor in bleaching events, and I'd call it a reasonable concern rather than a settled result. We do have one ultraviolet absorbing molecule in our skin, and its story is a little dark. When a structural protein called filaggrin breaks down in the outer layer of your skin, one of the products is urocanic acid. It absorbs ultraviolet B nicely, and when it does, it flips into a different geometric form, and that form is immunosuppressive. It damps down local immune surveillance in the skin, which is now thought to be part of why ultraviolet exposure contributes to skin cancer in more than one way at once. The light writes the mutation, and the same light produces a compound that tells your immune system to look away. Our best passive sunscreen has a side effect that helps the damage go unnoticed. The fish version has no side effect at all, and the fish didn't even build it. It ate it. We did eventually work out what was going on, and being what we're, put it in a bottle. There are commercial cosmetic ingredients on the market now built on chinarine extracted from red seaweed, sold as an ultraviolet A filter. You can buy the coral's solution at a cosmetics counter. That's the only route by which a human being gets this molecule into their skin. So that's the surface. Now go all the way to the other end. 1.5 miles down, in permanent darkness, at the bottom of the Pacific, there's water coming out of the seafloor at temperatures that would set your kitchen on fire. Hydrothermal vents were found in 1977 on the Galapagos Rift by scientists in the submersible Alvin, who were looking for warm water and geology and found an ecosystem instead. 2 years later, on the East Pacific Rise, they found the extreme version, black smokers, chimneys of precipitated mineral pumping out fluid at temperatures that have been measured above 700 degrees Fahrenheit. It doesn't boil, because at that depth, the pressure is far too high to allow it. The same force that makes the deep sea uninhabitable for our proteins is holding that water liquid. And a few inches away, the ocean is at about 35 degrees Fahrenheit, near freezing. That is the gradient. Hundreds of degrees, across the width of your hand. Animals live inside that gradient, with different ends of the same body sitting in wildly different water. Before we get to them, what does heat actually do to a person? Because this is one where the popular understanding is roughly right, and the details are worse than people think. Your core sits around 98.6 degrees Fahrenheit, with some normal variation. Push it to about 104, and you're into heat stroke territory, where the regulatory system has failed and the body is no longer holding the line. Past that, at 106 or 107, cells start to come apart in a cascade. Proteins denature. The gut lining becomes leaky, releasing bacterial products into the bloodstream, which triggers a body-wide inflammatory response. Clotting goes haywire. Muscle breaks down and floods the kidneys with debris. The margin between your normal operating temperature and catastrophic failure is about 8 degrees Fahrenheit. That's the whole range. Our defense is sweat, and it's a spectacular piece of equipment. Humans sweat more copiously than any other primate and among the most of any mammal, up to a couple of liters an hour in an acclimatized person working hard in the heat. Evaporating that water off your skin carries away an enormous quantity of energy, which is why a person can jog across an African afternoon that would drop most quadrupeds, except that the entire system depends on evaporation, and evaporation depends on the air being drier than your skin. Put a person in hot water, and the sweating apparatus becomes worthless in the most complete way imaginable. There's nowhere for the sweat to go. Meanwhile, water carries heat away from, or into, a body vastly faster than air does, so the transfer runs in whichever direction the water wants, at speed. This is why public health guidance on hot tubs is so unfussy and so strict about 104 degrees Fahrenheit and short exposures, and why people occasionally die in them. In water, your thermoregulation is essentially a suggestion. Now put yourself on a chimney wall where the fluid is well over 300. The animal that lives there is a polychaete worm called Alvinella pompeiana, which people call the Pompeii worm because of the fine gray-white fleece covering its back. That fleece is a dense mat of symbiotic bacteria that the worm cultivates on its own skin, in filaments held by specialized glands. And their role appears to include chemical detoxification and possibly nutrition. The worm builds a papery tube directly onto the outside of a black smoker chimney, in the zone where superheated fluid mixes with cold seawater. It sits in that tube with its tail down toward the hot end, and its plumed head sticking out into cooler water. Along the length of one worm, the temperature can change dramatically. The animal is straddling a thermal cliff. For about 30 years, nobody could agree on how hot it actually gets in there, and the argument was more interesting than either answer. Probes pushed into the tubes by submersible pilots kept coming back with high numbers, well past 140 degrees Fahrenheit, which would make this animal by far the most heat-tolerant complex organism known, and would push the theoretical ceiling for animal life upward. Meanwhile, the indirect biochemical evidence, the stability of the worm's own proteins measured in the lab, kept saying that shouldn't be possible. The problem was that nobody could get one alive to the surface. Bring a vent animal up without maintaining pressure, and it dies on the way. So every experiment was being done on a corpse. It took the development of pressure-maintaining sampling gear and a pressurized laboratory aquarium to settle it. A French team published the result in the journal PLOS One in 2013, having finally brought live Pompeii worms to the surface at pressure and then heated them under controlled conditions. 2 hours in the range around 125 degrees Fahrenheit killed them, with visible tissue damage and a full-blown cellular stress response. So the ceiling for this animal sits below that, not above it. The heroic numbers from the probes had been reading the water. The worm was sitting somewhere cooler inside its own tube. I want to make sure the correction doesn't read as a deflation, because the corrected number is still absurd. The same work put the worm's thermal optimum above 107 degrees Fahrenheit. Its preferred operating temperature is above the point where a human being is in fatal heat stroke. And a related species in the North Pacific, Paralvanella sulfancola, was put in a laboratory temperature gradient by Peter Gergis and Raymond Lee, and given a choice of where to sit. It chose the range between about 104 and 122 degrees Fahrenheit. Voluntarily. It tolerated up to about 130, an animal that prefers the temperature of a hot tub at its maximum legal setting and treats the temperature of a very hot cup of coffee as survivable, and does it while breathing water full of poison. How? Not with one clever molecule this time, with all of them. When researchers sequenced the transcriptome of the Pompeii worm and looked at the amino acid composition of its proteins as a whole, a signature came out that thermal biologists recognize immediately. Compared with related worms from ordinary temperatures, its proteins are shifted toward charged amino acids and away from uncharged polar ones. Charged residues form salt bridges, electrostatic clasps between different parts of a folded protein, and salt bridges are one of the main things holding a structure together when heat is trying to shake it apart. That bias, charged over polar, turns up again and again in heat-loving organisms, and it turns up across this animal's entire proteome. You cannot install that. It's thousands of proteins, each one edited, none individually dramatic, adding up to a body that holds together where ours would come apart. Its structural material got the same treatment. Collagen is the scaffolding protein of animal tissue, a triple helix, and it unwinds at a characteristic temperature that's uncomfortably close to normal body temperature in most animals. The Pompeii worm's collagen holds its shape at temperatures that would unwind the collagen of related worms entirely, achieved through differences in how the molecule is chemically modified after it's built. You have the emergency version of this. Everybody does. Heat shock proteins are among the most conserved things in biology, present in bacteria and in you, and they act as chaperones, grabbing proteins that are starting to unfold and helping them refold. When you run a fever, they're working. The Pompeii worm has them too, and it turns them on when it's pushed too far. Everybody carries the emergency kit. What differs is where the emergency starts, which leaves the last problem at a vent, and it's the one nobody thinks about because heat is more cinematic. Vent fluid is loaded with hydrogen sulfide, the rotten egg gas, That compound is the entire energy basis of the ecosystem down there, because there's no sunlight and no photosynthesis, and the bacteria that form the base of the food web make their living by oxidizing sulfide. Hydrogen sulfide is also a lethal poison, and it kills by essentially the same mechanism as cyanide. It binds to cytochrome C oxidase. It binds cyanocrine. It binds the enzyme at the end of the electron transport chain in your mitochondria and shuts down the final step of aerobic respiration. Your cells have oxygen. They just can't use it. Industrial hydrogen sulfide exposures kill workers in confined spaces every year, and part of what makes it so dangerous is that at high concentrations, it paralyzes your sense of smell almost instantly, so the warning disappears exactly when the danger peaks. You do have a defense at trace levels. Your mitochondria carry an enzyme that oxidizes small amounts of sulfide, and your gut needs it, because the bacteria in your colon produce sulfide constantly. Your body is quietly detoxifying this poison all day. The system is sized for micromolar amounts. Now meet Riftia pechiptila. The giant tube worm is the animal from every photograph of a vent. White tubes up to 6 or 7 feet long, topped with a blood-red plume. And the adult has no mouth, no gut, no anus. It doesn't eat anything ever. Inside its body is an organ called the trophosome, and the trophosome is a farm. It's packed with symbiotic bacteria, billions of them per gram, and those bacteria oxidize hydrogen sulfide to produce energy and use that energy to fix carbon dioxide into sugar. The worm is fed from the inside by tenants it acquired as a larva and then sealed in, which creates a delivery problem of remarkable awkwardness. The worm has to collect oxygen from the seawater and hydrogen sulfide from the vent fluid, and carry both in the same bloodstream to the same organ, without the sulfide poisoning its own mitochondria on the way, and without the two chemicals simply reacting with each other in transit. Its hemoglobin does it. Riftia carries giant extracellular hemoglobins, molecules far larger than the one in your red cells, floating free in its blood, and they have separate binding sites for oxygen and for sulfide. The sulfide is bound at its own site, held, transported, and handed over to the bacteria, while oxygen rides along on the standard iron chemistry at the same time. Bound sulfide isn't free sulfide, so it can't go and jam the worm's own respiratory enzymes. It's a molecule that carries fuel and poison in the same vehicle, keeps them apart, and delivers both to a colony of farmers living inside the animal's torso. And it works well enough that Riftia is one of the fastest-growing marine invertebrates ever measured. Work published in Nature in the 1990s recorded juveniles colonizing new vent surfaces and growing on the order of 2.5 feet in a year. Stack all of that up, and something starts to show through the individual stories. And it took me a while to see it. Almost none of these systems are organs. Look at what actually did the work. A repeating 3 amino acid unit that came from a stuttering pancreas gene. A small waste-adjacent molecule dissolved in muscle. A handful of amino acid substitutions on the outer surface of a protein that changed nothing about how it works and everything about how densely it can be packed. A binding curve tuned to react to acidity. A compound the animal doesn't even make, eaten and relocated. The two big structural exceptions are the icefish heart and the seal spleen, and both of those are enlargements of something that was already there. That's what evolution can afford. It edits. It duplicates a gene and lets the copy drift. It changes a few residues on a surface. It turns a knob on a molecule that already exists. Building a whole new organ from nothing is expensive and slow, and the record of these adaptations reads like a workshop with very good hands and a very limited parts bin. The second thing that shows through is that every single one of these has a bill attached, and I've tried to name the bill each time, because a system without a cost is usually a system somebody has described badly. The antifreeze fish carries ice inside its body that will never melt. The ice fish runs a circulatory system that fails if the water warms by a few degrees. The snailfish is locked out of the deepest quarter of the ocean by the very molecule that got it as far as it did. The Humboldt squid's oxygen delivery depends on the acidity of seawater, which is changing. The coral's sunscreen is supplied by tenants it evicts under stress. The vampire squid bought its oxygen budget by giving up the ability to chase anything. The Pompeii worm lives on a chimney that will eventually clog, collapse, or go cold. And when a vent dies, the whole neighborhood dies with it. The third thing is that none of it transfers. Take the fish's antifreeze protein and put it into a human organ bank, and it grows needles. Take the ice fish's cardiovascular design and give it to a warm-blooded animal, and the heart can't possibly keep up. Every one of these solutions is soldered to a specific temperature, a specific depth, a specific chemistry, and it's useless or actively harmful a short distance outside that envelope. And then there's the fourth thing, which is the one I find hardest to argue with. We solved every insult in this video, all of them, in roughly a century and a half. Cold water, the wetsuit, invented in the early 1950s, and the dry suit after it. A neoprene sheet with gas bubbles trapped in it, doing by insulation what the notothenoid does by chemistry. Oxygen on a dive, the demand regulator, the scuba set, and then closed-circuit rebreathers that scrub carbon dioxide and recycle the gas so a diver can stay down for hours. Nobody grew a bigger spleen. Somebody machined a valve. Pressure. Gas mixtures blended by the percent. Saturation systems. Decompression schedules calculated to the minute. Nobody grew a PS Elite. We got arithmetic, a gas blender, and a steel chamber. Salt. Reverse osmosis desalination, which does the ionocyte's job by brute force, pushing seawater through a membrane at pressure until fresh water comes out the other side. There are ships and cities running on it right now. Ultraviolet, chemical, and mineral sunscreen, sunglasses, shade cloth, and eventually an international treaty about the ozone layer, which is the only item on this list that involved fixing the sky. Heat, refrigeration, air conditioning, and fire suits. 6 insults, 6 answers, none of them written into a single gene. There's a difference in kind between the two approaches, and it goes in both directions, which is why I don't think either one is the winner. The animal's version is permanent, free to run, and it never gets left in the car. It works while the animal sleeps. It can't be lost, forgotten, or stolen. It also took millions of years. It only benefits that animal's own descendants, and it can't be revised when conditions change faster than a lineage can breed. Ours is removable. That's the whole trick and the whole weakness in one word. You can take the wetsuit off, which means it can also fail, be dropped, run out of air, or not be there. But because it's removable, it's also transferable. One person works out how to blend a hydrogen-helium mix in 1992, and every commercial diver on Earth has access to the knowledge inside a year. An Antarctic fish can't hand its antifreeze gene to a fish in the Arctic. The Arctic had to invent the whole thing over again from scratch, from a different gene, over millions of years, and arrive at almost exactly the same molecule. We invented it once and put it in ice cream. So why don't we have any of it? There's a version of this question that gets asked in a slightly wounded tone, as though the human body were a product that shipped with features missing. That framing is wrong at the level of how evolution works, and the correction is worth spelling out carefully, because it's the actual answer to the title. Natural selection doesn't reward preparation. It cannot. There's no forward-looking part of the process, no committee reviewing future risks, no mechanism by which a lineage acquires a defense against a threat it has never encountered. What actually happens is narrower than most people picture. A random variant appears in some individual because copying DNA is imperfect. If that variant happens to help in the environment the population is living in right now, its carriers leave slightly more descendants, and over enough generations the variant spreads. Every step of that requires the pressure to be present. Take away the pressure, and the variant is just noise, drifting, likely to disappear. An antifreeze glycoprotein is only useful to a fish that's meeting ice. In a fish that never meets ice, the same mutation is a metabolic cost with no return, and selection quietly discards it. Our ancestors never met these insults. That's the whole answer, and every branch of it holds up. We came from land vertebrates. The lineage that leads to us left the water something like 370 million years ago and never went back in any committed way. We're tropical and temperate in origin, terrestrial, air-breathing, and about as far from the deep sea as a vertebrate can get while still being on the same planet. Nobody in our ancestry sat under polar ice. Nobody lived below a few hundred feet of water. Nobody drank seawater as a way of life. Nobody spent their days in a lair with 2% oxygen. Nobody lived on the side of a hydrothermal chimney. Not once, not briefly, not for the 20 or 30,000 generations it would have taken for any of these systems to get off the ground. And here's what makes me confident that this is the real explanation rather than a comforting one. There's a control condition. There's one insult on this list that our ancestors did meet, in force, for a very long time. Ultraviolet light. Human ancestors spent millions of years in open equatorial Africa under some of the most intense ultraviolet exposure on the land surface of this planet. And what do we have? A superb, sophisticated, actively regulated pigment defense with a specialized cell type that manufactures it, a delivery system that hands the pigment to neighboring cells, a mechanism that positions it directly over the nucleus like a lid, and a whole feedback loop that produces more of it on exposure. On top of that, a repair crew for the damage that gets through, and a geographic gradient of pigmentation across human populations that tracks ultraviolet intensity closely enough to be one of the clearest signatures of selection anyone has found in our species. On the evidence, we're excellent at ultraviolet. Same story with heat. Incidentally, as long as the air is dry, our sweating capacity is close to the best in the mammal world. And combined with our upright posture, our reduced fur, and our long tendons, it makes a human being one of the most formidable long-distance movers in hot conditions on Earth. There are hunting traditions built entirely on the fact that a person can keep going in midday heat after an antelope has to stop. Our kidney is respectable for a large mammal with reliable access to fresh water. Our immune system is extraordinary. Our capacity to acclimatize to altitude, while it'll never rival a bar-headed goose, still lets people live permanently at 14,000 feet. Where our ancestors lived, we're superb. The gaps map insult by insult onto places they never went, and run the mirror, because the comparison only means something if it goes both ways. Put a Weddell seal on a savanna at noon. It has no sweat glands worth the name, it's wearing 4 inches of blubber, and it'll die of heat within hours. Put an ice fish in a temperate river, and it suffocates in water that a trout finds comfortable. Put a Hadal snailfish in a shallow bay, and its proteins, tuned to be loose enough to function under 800 atmospheres, become sloppy and unstable at surface pressure, which is why bringing one up alive is so difficult. Take the Pompeii worm off its chimney, and it has nowhere on Earth to go. Every one of these animals is a specialist, and specialists are defined as much by what they can't do as by what they can. There's one more piece of evidence, and it's sitting inside you right now. Go back through the list and count how many of these systems we have in a trace form. The diving reflex, present, weak, but unmistakably the same reflex. Blood shift into the chest under pressure, present. Spleen contraction on breath hold, present and measurable. Counteracting organic osmolytes, protecting proteins against urea, present running full-time in your kidney medulla. heat shock proteins, present and identical in function to the worms. A molecule in your skin that absorbs ultraviolet B, present. An enzyme that detoxifies hydrogen sulfide, present in your mitochondria. Trimethylamine N-oxide, circulating in your blood at this moment. Every single component is in the parts bin, all of it. The genes for chaperones, the transport proteins, the counteracting solute chemistry, the reflex wiring. What's missing isn't the toolkit. It is the assignment. Genes get switched on when there's a reason. Whole systems get built out when the pressure to build them is sitting on the population, generation after generation, killing the individuals who lack them. Remove the pressure and the components stay in the genome doing small, ordinary jobs, which is exactly where we find them in ourselves. A dive reflex that trims your pulse a little. A spleen that donates a cupful. A kidney medulla quietly running the same protein-stabilizing chemistry that a snailfish runs in its entire body. And the bajau are the proof that the machinery still responds. 1,000 years of diving for a living, which is nothing, an eye blink, and there's already a measurable, heritable change in the size of an organ. Give that population 100,000 years of the same life, and I wouldn't want to bet on where it stops. The process isn't switched off in us. It just hasn't been given anything to work on. There's an idea underneath all of this that I find hard to shake. A body is a document about a place. Every one of these systems is a sentence written by an environment, and if you know how to read them, the animal tells you where it's been. Find antifreeze glycoprotein in a fish, and you found a record of the Southern Ocean cooling tens of millions of years ago, written into a gene that used to make a digestive enzyme. Measure the trimethylamine oxide in a fish's muscle, and you can estimate the depth it lives at within a few hundred meters from chemistry alone. Look at the net surface charge of a mammal's myoglobin, and you can read how long its ancestors could hold their breath, including ancestors that have been extinct for millions of years and left nothing but a sequence. That's a startling thing to be able to do. These molecules are field notes, so read ours the same way. What does a human body say about the place its ancestors lived? It says, open country, a great deal of sun, dry heat, long distances covered on foot in daylight, fresh water reachable within a day, and air, always air. Never more than a few minutes without it, and never any reason to plan otherwise. It says a species that solved its temperature problem with water evaporating off bare skin, which only works in a dry atmosphere, which tells you the atmosphere was dry. It says a species that solved its ultraviolet problem with pigment, which only works if you can afford to be opaque, which tells you nobody needed to see through us. Every gap in the list from this video is a place our ancestors weren't. That's what I want to leave you with, and I think it changes how these animals should land. The temptation with a subject like this is to run it as a ranking. The seal beats the human. The snailfish beats the seal. Somewhere at the bottom of that table sits us, soft and slow and drowning in 3 minutes. And there's a certain type of video that ends right there because it's an easy note to hit. It's also wrong, and it wastes the actual finding. None of these animals is better at being alive than you are. Each of them is more precisely shaped to one narrow set of conditions than your, and precision of that kind is bought by giving things up. The ice fish gave up its ability to live anywhere warm. The vampire squid gave up hunting. The tube worm gave up its mouth. The Hadal snailfish gave up the surface so completely that surface pressure would leave its proteins too loose to work. The Pompeii worm gave up the entire ocean, except for a few square yards of unstable rock that will one day stop venting. Generalists give up peak performance. Specialists give up options. That's the trade everywhere, in every lineage, without exception. And both sides of it are still swimming around out there, which tells you neither answer is the correct one. Now, before this ends, I want to ask you something specific. because I've been chewing on it for weeks, and I have no idea what most people would say. If you could install exactly one of these systems in your own body, and you had to accept its bill, which one would you take? Not the fantasy version, the real version with the cost attached, the antifreeze, and the ice crystals that stay in your spleen for the rest of your life. The Seal's oxygen stores, and a spleen the size of a football, and blood so thick your doctor would panic. The tube worm's arrangement, and no mouth. The vampire squid's oxygen budget, and no ability to chase anything ever again. Put your answer down in the comments and tell me what you'd be giving up for it, because that second part is where it gets interesting, and it's where most people stall. I go through the comments on these, and I'll be arguing with a few of you. If you got this far, the like button does what it says, and subscribing means the next one turns up on its own. There's a lot more ocean. And here's where I want to land the whole thing. Every system in this video exists because something was trying to kill an ancestor, relentlessly, for a very long time, in a place that wouldn't let up. The antifreeze exists because of ice-touching gills. The oversized heart exists because a gene broke in the one ocean on Earth cold enough to make that survivable. The piezolite exists because 800 atmospheres was pulling proteins open. The sunscreen exists because a transparent animal can't afford to be dark. The heat-stable proteome exists because a chimney was pouring out water hot enough to cook. All of it was response, every piece of it, an answer to something that was already happening. And the reason your body carries none of these things is beautifully simple once you stop treating it as a shortcoming. Nothing in your ancestry ever stood in the cold under an ice shelf. Nothing ever descended past the depth where a lung is a liability. Nothing ever drank the sea, or sat in water with the oxygen stripped out of it, or held a position on the wall of a black smoker. The insult never arrived. So the answer was never written. You are not missing anything. You are carrying the complete record of somewhere else.